Growth characteristics of cancer cells. This covers the same competency as the previous class on proto-oncogene 10.1. So we are going to see something about growth characteristics of the cancer cells. ये पेन कैसे होगा? Yes, excuse me, just one. So the cancer is not a single disease, a large number of diseases involving various organs, and the common feature is uncontrolled, abnormal, excessive proliferation of the cells, resulting in production of abnormal cell mass. There are some Greek words, neoplasia, new growth, kericoma. It is actually means crab because the cancer cells, they sprawl in all directions like feet of a crab, movement of a crab, which is very abnormal. So these are a few things. But before we come to growth characteristics of these abnormal cells, let us outline some features regarding growth characteristics of a normal cell. The life starts from a fertilized ovum called zygote, and then there follow a chain of mitotic events, one cell into number of cells in successive cell divisions. And along with division, there is differentiation also. So from the progenitor cells are produced squamous cell, cuboidal cells, connective tissue के various types, all of you know it very well. So the life starting from zygote progresses through a chain of mitotic events, and all these events are regulated by certain stimuli, which not only regulate rate of the division, but differentiation into various types, as I mentioned, and finally, maintain differentiated state of the diverse cells. So, meticulously timed sequence of events. Even a mitotic division is under great control. There is replication in the S phase, mitosis in the M phase. There are long gap phases between the two. And as we discussed in the last class, I outlined once again, this is all under control of growth factors. More than 100 of them are known. So by a series of events are produced cyclins and cyclin-dependent kinases, which cause phosphorylation of multiple proteins, intracellular protein, which result in progression of the cell cycle. Okay, so it is all meticulously timed events. In cancer, what happens? This control is lost. There are certain other controls also which are lost. So we return to the normal cell. All of you know that the normal cell has got a finite lifespan that varies from cell to cell, but every cell having performed its duty, it dutifully quits. That is called apoptosis or the programmed cell death. So it is a sort of cellular suicide. It's a clean process. Various proteases, nucleases are activated and the cell itself dies through action of these enzymes. And then division, it is coordinated with physiological needs of the body as we saw. तो कैंसर के अंदर क्या होगा? Dysregulation of all these will occur. So the cell which becomes cancerous, हम उसको कहते हैं malignant transformation. The cancerous cell, it has been malignantly transformed. तो हम थोड़ा सा, now we are in a position to, having understood the normal cells, what happens, certain characteristics of the cancerous cells. First, mitogen independence. As we discussed in the last lecture, normal cells, they exhibit mitogen dependence. Just for example, growth factor, this will act with the receptor for growth factor by a chain of signaling events, cell cycle will be progressing. So the sum of the story is that mitogen, the growth factor, that is determining the progression of cell cycle. But in cancer cells, there is mitogen independence. The cell will divide even in absence of this mitogen. It does not need mitogen to undergo mitosis. And you can, just at the cost of repetition from the last lecture, some of the proto-oncogenes, they get converted to the uncontrolled overactive form called oncogene. But anyway, for today's lecture, mitogen independence is there exhibited by the cancer cells. So, abnormally high mitotic rate hoga. Second, they infinite lifespan is there. क्या मतलब इसका? They have lost capability for apoptosis. Unlike normal cells, which have got a finite lifespan, they have got infinite lifespan. That has something to do with telomerase that we will cover in the molecular biology, probably, or today is at the end of lecture also. I may talk something about that. So, lack of apoptosis is the second one. Third, encourage independence. Normal cells, they require an बेस ऑफ एक्स्ट्रा सेलुलर मैट्रिक्स जिसके ऊपर वो डिवाइड करते हैं. द एक्स्ट्रा सेलुलर मैट्रिक्स दैट डज नॉट प्रोड्यूस ओनली द इनर्ट बेस नहीं होता है, इट प्रोवाइड्स माइटोजेनिक स्टिमुलाई ऑल्सो. जैसे ग्रोथ फैक्टर्स हमने देखा था, दे प्रोवाइड द माइटोजेनिक स्टिमुलाई. द एक्स्ट्रा सेलुलर मैट्रिक्स ऑल्सो थ्रू सम प्रोटीन्स कॉल्ड इंटेग्रिन that provides mitogenic stimulus to the normal cell. So normal cells are encourage dependent. But और उसको अगर फॉर एग्जांपल सेल कल्चर में देखो, अगर आप ये सेल कल्चर में मैंने दिखाया है फाइब्रोब्लास्ट, they are forming a monolayer. They are actually anchored onto the solid base. But the cancer cells, they do not require any base, so they can divide, spread, sprawl in all direction. And coupled with this, I may talk another also, they lack contact inhibition. What is contact inhibition? Contact inhibition का मतलब अगर एक सेल डिवाइड हो रहा है, तो the neighboring cells of the same type, they inhibit each other's further division. All right? They inhibit further division by contact. कैंसर सेल में ये कॉन्टैक्ट इनिबिशन भी खत्म हो जाता है, so they are free from a solid anchorage independence. They are free from contact inhibition also. So no wonder, coupled with high mitotic rate, they spread, they sprawl in all direction. With the result, they form multi-layered structure. अगर ये कैंसर सेल होता तो कैंसर सेल में not only this, lot of other cells, multi-layered, disordered growth will be there. So this is what happens in cancers. So 4 characteristics we have discussed, but they are all interlinked. तो अभी तक हमने देखा requirement for mitogen, not in cancer cells, contact inhibition, not in cancer cell, encourage dependence, not in cancer cells, और independence, yes, in cancer cells. और हम थोड़ा सा देखने वाले हैं rest of the 3. So next one, cancer is a disease of genome. कैंसर के अंदर जेनेटिक एबेरेशन होंगे. कोई भी कैंसरस सेल्स है, उसके अंदर डीएनए के अंदर कुछ ना कुछ एबनॉर्मेलिटी होगी. दिस इज कॉल्ड जेनेटिक एबेरेशन. So cancer is a genetic disease, but it can be traced to certain permanent changes in DNA. But it is not an inherited disease. Is it looking paradoxical? Is it looking paradoxical? Opposite statements, no, they are not. So I leave it to you, self-directed learning. It is a disease of genome, but it's not an inherited disease. Okay, so as we are, I may hint you also, कि cancer में कोई, so okay, we discuss with this slide. Philadelphia chromosome. This is found in chronic myelogenous leukemia. There is a gene translocation. So the chromosome number 22 इसका shortening हो जाती है. Now if you see a patient of CML, only the cancer cells will have Philadelphia chromosome generated as a result of chromosomal translocation. But the normal cells of the same patient will not have this Philadelphia chromosome. इसका क्या मतलब हुआ? Some somatic cell or some group of somatic cells underwent this mutation and that was propagated further. Alright, so genetic aberration, Burkitt lymphoma, again there is a gene translocation, neurofibromatosis, a RAS protein, a G protein, उसके अंदर कुछ प्रॉब्लम आ जाती है and again the same thing. Retinoblastoma, there is an anti-oncogene called RB gene, retinoblastoma gene, इसके अंदर कुछ म्यूटेशन हो जाता है and I may repeat, only the cancerous transformation wale cells, the cells which have been transformed, which have undergone malignant transformation, will have these mutations. Normal somatic cells will not have. So that provides evidence that this is because of mutation, genetic abnormality in some somatic cell. So this is actually, So all these we have discussed so far. 2 or 3 are remaining. This also, two M's, metastasis and monoclonal origin. What do we mean by that? Monoclonal origin. तो monoclonal origin का हमने already hint कर दिया. A somatic cell or a group of cell उनके division से होता है. Supposing एक somatic cell, let us assume एक या दो somatic cell हुए, they will divide into hundreds of cells. So all these cells having originated from the same cell, we can say that they have got an monoclonal origin. So they belong to the same clone, same group. So this is monoclonal origin और उसका proof हम already दे चुके हैं. Chronic myelogenous leukemia में क्या होता है? All the cancerous cells only are having Philadelphia chromosome. So that, as we discussed, provides evidence that origin is monoclonal. Next M, metastasis. Metastasis means, this slide, colonizing far off tissues. The cancer cells are carried to distant tissues by blood and lymph, where they form secondary growths which are called metastasis. Why they are able to do so? Because we had discussed there is no anchorage dependence. They are not anchored and each cell is showing a disordered growth pattern. So they are not tightly bound to the committed to the base, the original site. So they can break loose and move to various sites. Actually, colonizing far off tissues, इसके लिए जरूरी होता है 3 points. Number 1, it should be loosely bound to its original site. Number 2, it should be able to penetrate the basement membrane. And the third and the most important, it should have ability to survive in the new location, in the newer location, in the newer surroundings. The normal cells, they more or less, they cannot find this capacity. किसी पौधे को जड़ से उखाड़ कर फेंक दो. The weeds, etc., they can grow at any size, but not our pampered plants, etc. All right. So we have discussed the last, we said anaplasia. Anaplasia को de-differentiation भी बोलते हैं, which we are going to see now. So this slide, same thing, but one additional term, aneuploidy, change chromosome number, amplification, translocations, etc. So all this we have done. Now we are going to talk last characteristics in this context, anaplasia. Anaplasia का क्या मतलब होता है? I will return to this slide, please. Sorry. Yeah. Anaplasia. You people know very well that cell जब divide हुए, at the beginning we said they differentiate into various form. अगर हम epithelial cell कोई consider करें, squamous, cuboidal, columnar. So this is called differentiation. कैंसर सेल्स के अंदर डी डिफरेंशिएशन उल्टा हो जाता है. The differentiated cell assume characteristics of their progenitor. They become more and more like embryonic cell. They assume a rounded appearance, the nuclear cytoplasm ratio that increases, और ये जो differentiated appearance है वो धीरे धीरे लूज होती जाती है. So they assume shape and characteristics of their progenitor cell, the embryonic cell. And like embryonic cells, they can differentiate into further, later on into various cell types. We are going to stop at that. So differentiation, cancer cell lose the appearance, undifferentiated appearance, इसके दो characters जहाँ बताए हैं. So anaplasia, so assume feature of immature cell, loss of structural and functional differentiation of the mature cell that occurs. So these were all the characteristics of the cancer cells. Biochemical characteristics, of course, you can extrapolate. ज्यादा डिवीजन हो रहा है तो increase synthesis of DNA, RNA, protein synthesis, ज्यादा degradation होगा, negative nitrogen balance. I just give you a problem here. ल्यूकीमिया के पेशेंट को एलोप्यूरिनॉल क्यों दिया जाता है? ल्यूकीमिया में ज्यादा सेल प्रोड्यूस हो रही है, ज्यादा सेल ब्रेक डाउन भी होंगे. यूरिन डिग्रेडेशन से क्या बनेगा? यूरिक एसिड. तो हाइपर यूरिसीमिया होगा, उसको ट्रीट करने के लिए एलोप्यूरिनॉल होगा. इफ यू हैव नॉट अंडरस्टूड, आई हैव गॉन वेरी फास्ट. व्हाई एलोप्यूरिनॉल is given, a suicide inhibitor of xanthine oxidase is given to leukemia patient. So of course, zyada honge increase rate of this cycle, but there are various aberration. Cancer cell itself is an abnormality. तो ये जरूरी नहीं ये सब हो. Now many cancer cells उसके अंदर TCA gets curtailed. Okay, TCA curtail हो जाता है and the energy is derived from some other sources. So secretion of certain proteases, this is all descriptive types. You can just take down if you want these ones. So synthesis of fetal proteins, tumor marker का जब आप करोगे तो ये सारा आएगा. All right. So this table summarizes everything. The most important points. इसको जरा highlight मैं करना चाहूँगा. Abnormal proteins are synthesized. जैसे अल्फा फीटोप्रोटीन, that is synthesized by the fetal cell. So would you speak something about it?
Embryonic liver cells may it is produced, which stops its expression after the amount of gestation period.
Yes. So after the gestation period, expression of alpha fetoprotein, we call abbreviated AFP, it stops. हमारी तुम्हारी बॉडी में नहीं होता, but cancerous cells they start expressing this abnormal protein, fetal protein. And they are important in diagnosis and prognosis of cancer and called tumor concept. All right. अब जो मैंने ये बात की थी टीलोमेरेज की, ये टीलोमेरेज क्या होते हैं? They are concerned with immortality somehow. Germline cells express an enzyme called telomerase and therefore immortal. Somatic cell, hamare cell, they do not express telomerase and therefore they have got a finite lifespan. Even अगर आप सेल कल्चर में भी करते हैं ना, एक बच्चे का सेल कल्चर करो और एक elderly person ka, the child cell will undergo more cell divisions before it dies. Elderly ka ek ya do ke baad very soon it will perish. So telomerase is not expressed, so apoptosis hota hai. The cancer cell de-represses its telomerase. This portion shows how telomerase acts. I am going to omit. ये आपको थोड़ा सा ये इस फिगर को, this is advanced learning concept at the moment, but you will learn it in molecular biology. There are two strands leading and lagging. Lagging stand by successive rounds of replication, it undergoes shortening. This is called telomerase, telomer erosion, means शॉर्टनिंग होती जाती है. इसका क्या रीजन है? जैसे प्राइमर फॉर्मेशन हुआ यहां पर, प्राइमर इज removed and even if the primer is formed at the end of the chromosome, इसका रिमूवल थोड़ा सा गैप क्रिएट कर देगा. तो टीलोमेरेज क्या करता है? बाय अ सक्सेसिव ऑफ इवेंट्स इट फिल्स दिस गैप. बस इतना ही अभी काफी रखते हैं. दिस इज व्हाट टीलोमेरेज डज. So the problem, lagging strand is incompletely regulated. Solution, telomerase elongates and solve this problem. So telomerase solve this problem by preventing telomere erosion. Forget about this. Telomerase retard the cell death. Cancer cell के अंदर telomerase is de-repressed, so apoptosis, अजर अमर हो जाते हैं ये. They become immortal. Okay, like Greek gods, etc., Apollo, etc., they were all immortal. Maybe they express more telomerase. So jokes apart, I can. All right. So with this, we have compared properties of cancer cells with the normal cell. You must know characteristics of normal cell. I'm sure in histology you have been doing all that. Okay, thank you so much.